All right, in this lecture, I'm going to be talking about membrane transport. And this information corresponds to what you could find in your book, chapters 9, section 10. There are two major types of membrane transport. There is, sorry, let me get my laser pointer. Here we go. There is passive transport, which is moving something across the membrane that does not require the use of energy, and things are going to be moving with their concentration gradient, which is why it doesn't require any energy to have that happen. And then there is active transport, which is where you're going to be moving some type of molecules against their concentration gradient. That means going from an area of low concentration to an area of high concentration. So in order to move something against the current, essentially, you're going to have to put energy into that for it to go. As far as passive transport is concerned, there are two different types of passive transport. There is simple diffusion. This is where we have those very small nonpolar molecules that can just simply diffuse through the membrane. And then you have facilitated diffusion. And this is where you're going to have an integral membrane protein help you move a molecule from an area of high concentration to an area of low concentration. Typically, though, this molecule is either going to be too large, charged, or polar, which is why it can't use simple diffusion, because it'll never make it into this hydrophobic space of the membrane. Okay. So there's simple and facilitated diffusion. Those are both passive types of transport. And then as far as active transport is concerned, we also have primary and secondary active transport. So primary active transport requires the energy source of either ATP or light as its energy source to get the reaction moving forward, or to pull something from lower concentration gradient into a higher concentration zone. And then secondary active transport, again, moves something against its concentration gradient, but uses the help of ions to facilitate that movement. So instead of like breaking a molecule of ATP for energy, it's going to use an ion concentration gradient to help move something. And we'll see examples in a second of all of these. Okay. So let's start with passive transport. Look a little more deeper dive for passive transport. This is a slide from a previous lecture, so this is refresh, hopefully, or at least reiteration. All right, so we can have small nonpolar compounds like carbon dioxide, oxygen, nitric oxide, NO. Those can all easily move across a membrane with their concentration gradient. So from an area of high concentration to low concentration. But again, charged or large molecules cannot just simply diffuse through the membrane. So this is the only way to have some type of simple diffusion is if you have a small nonpolar molecule. The second type of passive transport was the facilitated diffusion. So this is where you have to have an integral membrane protein. Remember, that is a protein that spans both the inner and outer leaflets of the membrane. You'll need this protein to help move the molecule from the area of high concentration to the area of low concentration. Okay, so you can see kind of as it goes down. This requires no energy from the cell, but it just needs the help of this transporter to do that. You could call this type of a transporter a uniporter, uni meaning one, and porter for kind of transporter. So this type of transporter is only moving one molecule across the membrane, so you can call it a uniporter. So what are some examples of some facilitated diffusion? So I think water, at least in my brain, has always been a big thing of like, why would water, a very polar molecule, be able to get in that super hydrophobic region of the cell? And it was in 2003 when Peter Agri and Roderick McKinnon won a Nobel Prize for their discoveries concerning channels in the cell. But Peter Agri, specifically, he discovered water channels. So we're going to look a little bit more we're going to hear from him about aquaporins, that's what he called them, and learn more about these water channels that help facilitate the movement of water across the membrane.
My name is Peter Augrey. I'm director of the Johns Hopkins Malaria Research Institute at the Johns Hopkins Bloomberg School of Public Health, Baltimore, Maryland, USA. Some ways I think I'd say research chose me. I was a student in medicine at Johns Hopkins back in the 1970s, hoping to do some research relevant to the diseases of the third world. I was involved because of the humanitarian interest. I guess there was a threshold I must have passed where I became a scientist. It was not a one-way street at which I would end. It was science from here on. Being from a large family, I'm used to talking to people, sharing information, getting ideas. And I talked to scientists. I talked to dozens of scientists. Explained that we had a new protein. We thought it was a channel protein from the red cell membranes. And we always got the same response. Red cells don't have channels. You must be wrong. Until I finally talked to a colleague, John Parker, at the University of North Carolina at Chapel Hill, a very dear friend, a hematologist, a blood specialist, who was also a physiologist. And he immediately recognized red cells, renal tubules, roots of plants. These tissues have one shared property. They're highly permeated by water. Maybe this is the long sought water channel. It was before our first public lecture, my postdoctoral fellow, Greg Preston, wonderful fellow, charming young man. We were all having lunch, but he had to give the lecture, so he was the only one not to have beer. So we were thinking we need a name, a functional name for these water channel proteins. And there were 6 of us or 7 of us at lunch. And no one remembers exactly where the name came, but we were thinking of names like maybe hydropontin or hydrophorin. Someone else thought maybe aquaforin, or the Greek aquaporin. That's it. Everybody said, that's it, aquaporin. The generation of pure water is theoretically possible with the aquaporins because this is a water selective filter. Only water can go through this filter. The engineering of this, though, has some technical difficulties because the native membranes are very tiny, and so only nanoscale water purification can occur. A lot of individuals got very excited, attention from around the world, even a Nobel Prize. But, I honestly don't see myself as Einstein reincarnate. I see myself more as Huckleberry Finn, a happy-go-lucky individual with a good team of scientists and friends looking for adventure. And boy, did we find one.
All right, so that was Peter Augrey himself talking
My name
about
is Peter Augrey.
Of his discoveries and information about the history of discovering the aquaporins. All right, so let's look at the structure of an aquaporin. Again, they were researching red blood cells, and they're very porous, right? They can filter water, the flow of water very easily in red blood cells. And so this is what the structure of an aquaporin looks like. It is a homotetramer, meaning that homo meaning the same, tetramer meaning 4. So there are 4 subunits of the same exact polypeptide sequence. It is an integral membrane protein, meaning again, it spans both the inner and outer leaflet, so it's the whole way through the membrane. It is found almost in all living organisms, these aquaporins that people said you must be crazy, that things like this don't exist. They do, and it makes so much more sense now how water can easily and quickly pass through the membranes. In humans alone, there are 10 different isoforms. Isoforms, that can mean a couple things. One thing, when you hear the word isoform, think about small modifications in the amino acid sequence. Still has the same overall function as a protein, but maybe just a few different amino acid mutations. And typically those isoforms are expressed in different types of cells. So your kidneys filter a lot of stuff and have a lot of aquaporins in their cells, just as your red blood cells. So the aquaporins that are in your red blood cells are slightly different than the aquaporins that might be in your kidney cells. And they filter water, or they allow water in and out at 3 billion water molecules per second per pore. So that'd be 4 total pores. You can get 12 billion water molecules per second to pass the membrane to help with osmotic pressure and whatnot. Inside that pore, if we take a little closer look, water travels through in a single line. This is very selective for only water. The size of the pore only allows a very small water molecule in. If you were trying to think of maybe like a big chloride ion, chloride is number 35 on the periodic table, if I'm not mistaken, and oxygen is only element number 8. So think that's a lot more electrons, that's a lot more protons, and so chloride's going to be a lot bigger molecule than a water molecule will be. So just the pore itself will select against things like a chloride ion just based on size alone. So if you get something very small, small enough that is similar to the size of a water molecule, along this pore, the channel inside the pore that the water molecules are filing through in a single file line, you will find different residues, amino acids inside the pore that hold water in a certain orientation. So as it's coming into the pore, there's a central part inside the pore where a water molecule does a flip. So when we're coming down the pore, towards the top of the pore, it's slightly negatively charged. So the partial positive hydrogens are always facing towards the top of the pore just due to electrostatic interactions. And then there are 2 conserved asparagine residues right in the center of the pore. And that's actually where water does a little flipping motion. And then the hydrogens now end up pointing down towards the bottom of the pore as they're coming through, because there's, again, partial negative charges lining the pore here. So really fascinating chemistry, and just a small, simple thing like an aquaporin. We could talk a lot about the structure and the function and how water actually funnels through. So I have one more video that kind of sums up quickly the functions and the findings of aquaporins, and it's a claymation stop motion movie. So enjoy.
Aquaporins were first discovered by a team of researchers led by Peter Ager at Johns Hopkins University in the early 90s. Peter's team was studying membrane proteins in red blood cells. In the process of their research, they identified a protein that they thought was a water channel. To test this hypothesis, they inserted the protein into the cell membrane of frog eggs and then dropped the eggs into water. As water entered the egg cells through the water channels, the modified eggs swelled up and burst open, confirming their hypothesis. Aquaporins are integral membrane proteins that allow water molecules to passively move from one side of the membrane to the other. In this animation, a red aquaporin subunit is seen spanning a pink membrane. The blue and white represent the oxygen and 2 hydrogens in water, the yellow is chloride, and the purple is potassium. Watch how only water is able to pass through the aquaporin and down its concentration gradient. Now let's take a closer look at the structure of the aquaporin that gives rise to its unique function. Each aquaporin subunit is formed by 6 alpha helices bundled together. The inside of a channel is lined with negatively charged residues that allow for hydrogen bonding between the water molecule and the protein until the molecule reaches the center of the protein, where it is flipped by the positive charge around 2 asparagines. This change in orientation allows only water molecules to fit through the aquaporin, making it a highly specific channel. In this animation, the white negative signs represent the negatively charged lining of an aquaporin channel, and the purple plus sign represents the positively charged field around the 2 asparagines in the middle of the channel.
Right, so that's a really good little summary video. Again, I'll post these videos separately if you want to go back and watch them if you're studying or taking additional notes. Okay, so that's enough about about passive transport. So passive, remember, requires no energy, and it's moving things with their concentration gradient, either passive diffusion, which just means it can go through the membrane by itself, or via facilitated diffusion, which needs the help of an integral membrane protein to move that molecule with its concentration gradient. So from going from high to low concentration. So now let's look at active transport. So active transport requires the input of energy to move something against its concentration gradient. And again, there is primary and secondary active transport. Primary active transport either requires the use of ATP, so this first cartoon representation of an active transport is using ATP to move some cations against their concentration gradients. And then secondary active transport uses the couples moving something against its concentration gradient with moving an ion with its concentration gradient. So you're going to move two things at once. One's going to move with the concentration gradient, and that's going to be the ion that's going to help drive and create the energy to move something against its concentration gradient, which in this case might be glucose. Okay, a couple other buzzwords we can see here is there's an anti-porter and a sim-porter. Anti means against or maybe opposite, and then sim would mean together. So an anti-porter would be a transporter that moves two things opposite of each other. So the sodium-potassium channel is an example of an anti-porter because it's going to move potassium inside, yet sodium outside of the cell. And then a sim-porter is something that moves two things together. So sim meaning, together as one. So that would be an example of this glucose sim-porter here, where we're going to couple moving sodium with its concentration gradient to coupling the movement of glucose against its concentration gradient. Okay, so again, moving things against concentration gradients, let's just take a look. So here's the extracellular components. Sodium is at 145 millimolar, and internally, sodium is only at maybe 10 millimolar. All right, so, and then you can notice that we're going to move sodium from a 10 millimolar concentration, very low concentration, outside of the cell to a very high extracellular concentration of about 145 millimolar, right? And potassium is the same thing. You're moving it from an area of low concentration, so 5 millimolar, to an area of higher concentration, which is inside the cell, which is about 140 millimolar concentration. And then the sodium-potassium pump is responsible for helping to repolarize all of our neuron cells. So I have another video that goes into more in depth about the sodium-potassium pump. And it also talks about some of the ion-gated channels that open and close based on the ion concentration, either externally or internally in the cell, which all has to deal with you can really talk about the neurons, right? Moving a signal down from one side of the neuron to the other side. So let's watch this video, and then we're almost done.
Neurons in our nervous system use electrical impulses to transmit information throughout our bodies. This process is mediated by membrane proteins called voltage-gated ion channels. Voltage-gated ion channels open and close in reaction to changes in membrane voltage or membrane potential. A cell membrane potential is generated by concentration gradients of positively charged sodium and potassium ions across the membrane. When a neuron is at rest, its ion channels are closed, and the overall concentrations of ions and proteins create a higher potential on the outside and a lower potential on the inside. This state is referred to as resting potential, and the membrane is said to be polarized. To transmit a signal, the neuron creates an action potential which travels down the axon. The action potential is mediated by the opening of the voltage-gated sodium channels, which allows sodium to enter the neuron. This depolarizes the membrane in that local area. Neurons then repolarize the membrane in several ways. First, voltage-gated sodium channels close and become inactivated. Then, to counteract the positive charge of the sodium ions, potassium channels open to allow potassium ions to exit, lowering the voltage across the membrane. In this way, the signal travels along the neuron. Sodium channels open and trigger one another to generate action potentials, and potassium channels prime the neuron for the next signal by allowing potassium to exit. After the signal has finished, the locations of ions have been reversed. Sodium concentration is high on the inside, and potassium concentration is high on the outside. The sodium and potassium pump restores the sodium and potassium gradients, making the cell ready to send another signal. With each pumping cycle, this transport protein moves 2 potassium ions back into the cell and 3 sodium ions out of the cell. Because the pump moves these ions against their concentration gradients, it requires the energy of 1 ATP molecule for each cycle. This molecular machine comprises 3 chains, alpha, beta, and gamma. Within the alpha chain, 3 domains, A, P, and N, form the core engine of the pump. The pumping cycle occurs in several steps. With ATP bound in the N site and the transmembrane region open into the cell, the pump binds 3 sodium ions from the cytoplasm. This causes the N, P, and A sites to shift closer together. With the help of a magnesium cofactor, the P site is phosphorylated and ADP is cleaved off. This leads to a conformational change that exposes the sodium ions to the outside. The sodium ions can then be released, one after another, into the extracellular space. In this ion empty state, the pump allows the entry of 2 potassium ions. This causes dephosphorylation of the pump followed by closing of the extracellular gate and allows the N domain to accept ATP again. As a result of ATP binding, the intracellular gate opens, the potassium ions are released into the cytoplasm, and the cycle can start again. The sodium-potassium pump is a key protein that works to maintain the sodium-potassium gradients and intra and extracellular potential differences necessary for neuronal signaling. Roughly a third of the ATP made daily by our cells is spent to power this molecular machine.
All right, so that was an example of primary active transport, which is again the sodium potassium pump. And what's crazy to think of is that chemistry is occurring in your body all the time. Even when I'm talking right now, neurons are controlling my muscles, contractions in my face to help make the sounds come out of my mouth, the sounds I want them to make words. And think about all those action potentials that have to be made, and the cell, my neurons get depolarized, and then they have to repolarize. So that sodium potassium pump has to move back across the concentration gradients of sodium and potassium ions into their right spaces, so my brain can fire again, so I can say another word. It's just fascinating to think of your body working at that type of a level, at that kind of macroscopic scale. Okay, so to recap, there are 2 major types of membrane transport. There's passive, which is moving something from high to low concentration, so with its gradient. And then there's active, which is moving something from low to high, so something against its concentration gradient. And in order to do that, it's going to require the input of energy. Okay? As far as passive is concerned, there's simple diffusion, which is just molecules moving across the membrane with no help from anything whatsoever. And there's facilitated diffusion, which would use an integral membrane protein to help move a molecule with its concentration gradient, kind of like the aquaporins that Peter Agre founded. And then there's active transport. This is moving something against its concentration gradient. Primary active transport is going to couple that movement with the energy source ATP. And secondary active transport is going to couple that movement with the movement of an ion with its concentration gradient. Okay, the last thing I wanted to show you is this fun video is what I think of. It kind of also is like why that opening slide was a picture of a subway station. But I sometimes think of membrane as the cell, as like people getting in and off of a cart. So I found this really busy train station, and this is kind of the cell is what I think of. And then the doors are going to be like the gates that are going to let things come in and out. And so your cells are constantly being bombarded with things all the time that are trying to get in and things that are trying to come out. And so you can see, like as the doors open, a flood of people or molecules are now coming out into the extracellular space. And when they're done, things are rushing in. So I don't know. I think this is kind of a real world idea of what it could look like. Okay, so with that said, what are the takeaways from this lecture? What should you know when you go? You should be able to provide a definition for passive transport, active transport, simple or facilitated diffusion. Explain what primary versus secondary active transport is, and then talk about or draw or sketch an example of a uniporter, a sim porter, or an anti-porter. You should be able to explain how aquaporins work and how the sodium potassium pump works. So what are those steps by which you can get 3 sodiums out and 2 potassiums in, and where is that coupled with it, the hydrolysis of ATP? And then provide a real life example from this lecture of a uniporter, an antiporter, and a sim porter. And that would be the end.